Welcome, everybody, back to the podcast. Today, I have a very special guest to help me answer some of the harder questions around COVID vaccine technology. Because as you know, I'm an epidemiologist, a numbers guy, but I'm not a lab guy. And so I brought in the heavy guns. To help me out is a professor friend from Carleton University, and I'm going to pronounce his name the way I think it should be pronounced, and he can correct me afterwards. Dr. Erling. Is that right, Erling?
Very good. Maybe a little softer. The Norwegians would say it softer than that, but it's okay.
So tell us about yourself, Erling. What qualifies you as a vaccinologist and virologist?
Okay. Well, actually, I started out in science, I guess, in '76 when I started my university career. I'm very fascinated with every aspect of science, from microbiology right through to stargazing. So I came to it naturally. I was given a telescope and a microscope when I was 10 years old. So,
and you knew the difference at that point. You can turn them around and
one looks up, one looks down. Properly, my mother gave me the microscope, and being a good son, I followed my mother's lead and went with microscope work and not the telescope work. That my father gave me the telescope. Now, from '76, I actually got some interesting summer jobs, actually working on geochemical technology. I was looking at uranium and water samples being sent to us from all over Canada, usually up in the north. And in doing that work, it was geology, a fairly dirty industry, but there you go. And I was offered a job the following, 2 years later, with Parks Canada, very much looking at the environment and how we protect the environment. And that sort of was more down my bailiwick. Was doing a BSc in microbiology actually at the time, and very much got into environmental studies and how we can prevent the disasters in the environment that were occurring. We were using pesticides, bolus. How could we reduce the amount of pesticides used? So I did a master's degree at Institut Armand-Franpier in 1980 to 82, very much looking at how we could use insect viruses to control insect populations. Insect viruses are very specific to the insect they infect. So it wouldn't hit the non-target species, like the predators or fish or whatever in the environment, and reduce the amount of chemicals that were needed to be used to get what we called an economic feasible level. So bringing economics in, my first time into economics, not the last.
The fake science of economics. Sorry, economists.
Yeah. And then basically went from there, insect viruses, got into virology. We had some kids north of Montreal that came down with California syphilitis, a virus transmitted by mosquitoes. We don't even talk about this all that much anymore, but it's still around us, out in the woods. Stay out of the woods.
I've never heard of it.
Yeah, no, I know. I mean, it's amazing, actually. We have cases in Ontario and Quebec every year, but just low incidence, fortunately. But it's something transmitted by mosquitoes. And actually, we were able to isolate the virus in mosquitoes that overwintered in little ponds in just north of Montreal. So I took that virus and brought it down to Alabama and started to clone and sequence to look at what's going on with the variation in these. Why did we have kids coming now with severe encephalitis when we knew it was there and wasn't causing severe encephalitis in others? So was there a genetic variant or reassortment? That's what was going on in those days. We were looking at reassortments between different strains of the virus. And then it sort of brought me into doing my PhD on how viruses replicate and how we can interfere with their replication. I did that at the University of Ottawa. After coming back from Alabama, I decided to explore the use of genetics a little bit more for a better understanding of virology, and worked on how viruses generate their own defective versions of themselves and how that controls its own replication. Fascinating study. Hardly anyone understands it, to be honest.
I don't.
Well, hey, we can have a beer and I'll have a good talk to you about it.
I'll look forward to those days.
Yeah. And then, HIV hit, middle 80s. And I was looking for a postdoc. My wife, brand new wife, came home just after our wedding and say, oh, guess what? They're moving me to England. So I said, well, I guess I better get a job in England. So I toured around. I had fortunately convinced the Dean of Medicine at the University of Ottawa that I should be sent to this meeting in France. He thought I was a medical student at the time. I didn't See, it wasn't.
Didn't correct him.
Yes. So he sent me there. And then when he got back, he realized I was a PhD student, not an MD student. But he said, I'll pay for it anyways. So great. And toured around several labs in the UK and was offered a position by Fred Brown. Fred Brown is known for the use of peptides as a vaccine approach. He had his fame with foot and mouth disease virus. He worked at the time with Wellcome Laboratories, Wellcome Research Labs, which is now part of GlaxoSmithKline. But in those days, it was still owned by the Wellcome Trust,
Yeah.
which is very much the largest non-government funding agency in Europe. So it was great. I had all the resources of a company. I was funded by the UK Medical Research Council and very much supported in the guise of the trust. So it was a fantastic 5 years to work in an amazing environment. Worked on HIV vaccine development. We developed an attenuated vaccine approach, explored that in non-human primates. So very much involved in the preclinical testing of vaccines. That's how I got my creds in vaccines. It was very much, that was from 1989 onwards. So once I got my PhD on understanding how viruses replicate, I very much got into the vaccine development field. From the from the idea generation to the preclinical testing, almost to the point of clinical trials, when I was continually saying we've got to look at safety of attenuated HIV vaccines. We can't go there without really looking at the long-term impact on the youth, infants, etc. And my apprehension came to pass. We found out that giving this type of approach to very, very young monkeys would actually induce AIDS-like symptoms in those monkeys. So it put a hold on trying clinical trials with this approach. Came back to Canada in 1994, joined the Laboratory Centre for Disease Control, which is now part of the Public Health Agency of Canada. So put my public health hat on, but very much still in a research and development environment. But at that time, we were closely associated with the regulators in Health Canada. So I, on one side, would be working with companies, and the other side would be working with the regulators, providing them with science advice on what was coming through. So it was very much a heady environment of looking at what we could do and supporting the regulators on how they had to look at these products. So I've, during that time, developed a good understanding of how products are regulated and the apprehensions and very much the looking at the safety and efficacy of any approach, vaccines being one of them. Then at one point ended up going back to the Public Health Agency in about 2014 because of the Ebola situation, and was helping the Public Health Agency in regards to science advice on how to get that vaccine developed in Winnipeg, which was this VSV, vesicular stomatitis virus, generated product that had been developed years beforehand. And I actually evaluated the grant, or not the grants, but the papers going to Journal of Virology and others from that group, and really supporting their initiative of, yes, take that approach and go forward. However, as we'll understand, it is not an overnight enterprise to get a vaccine concept into clinical trials and actually getting into the marketplace. It takes major money. We're talking about billions of dollars and major, not only financial investment, but political investment. So for Ebola, it held back for a little bit. Public Health Agency worked with a small company. That didn't work out. Merck got involved, and then boom, they got their vaccine. They could try it in 2016 in West Africa. If we'd had that vaccine 5 years beforehand, which we could have, maybe those people in West Africa would have been protected. So I look at vaccine development in two sides. Yes, it takes a long time when there isn't the political support or the sense of urgency. But in this situation with COVID vaccines, there is that support. So those timelines have shortened because we have the finances, we have the political support.
Money makes magical things happen.
Exactly. And it's not that the safety wasn't done. They have done those studies. The questions went out. I asked people at the NIH when they jumped forward with Moderna, have you looked at those aspects? Are you looking at the potential adverse events? And they were. They didn't do this blindly at all. So I'm convinced that the right people were in the right place at the right time.
Excellent.
No.
So you are definitely the guy to talk to today.
Okay.
So as a a prelude to what we're going to talk about, I sent a link out to the world for people to submit questions about the COVID vaccine for us to attempt to answer. And I say attempt because we won't have all the answers, but we'll have probably pretty good guesses what the answers might be. Keeping in mind that neither one of us is a medical doctor. I know you're not one, right? I'm definitely not one. So we can't offer medical advice. That's between you and your clinician. Before we get to the questions, though, I wanted to ask you your opinion on this new plan between Oxford AstraZeneca and the Russians to do a combined test of their vaccine candidates. So people don't know, Oxford and AstraZeneca have a viral vector COVID vaccine, which has some questionable issues associated with their recent trial. And the Russians have a similar vaccine called the Sputnik V. And the proposal is to do them both together. Could you explain how that works and why it might be a good idea?
Well, it's very interesting, actually. When we look at vaccine approaches, the one is that you use one vaccine and then boost with the same vaccine in the future. And you see that, with hepatitis B vaccine or the A-B combination now, Twinrix. You boost with the same one. But there's other philosophies. That is that if you actually take the sequence you're interested in, so in this case, the spike protein, and you put it in one approach, this would be the chimpanzee adenovirus that AstraZeneca and Oxford are using. Put that in as the initial vaccination, and then come in with a different viral vector, DNA, whatever, or protein, and give that as the boost that you'll actually induce
3 weeks later, so
2-3 weeks later, maybe a month later. Then you actually will induce better immune responses against the thing you want to get an immune response. You won't necessarily boost the immune response to the vector. And this is the big problem we have with using the human adenovirus vectors as the first prime, is that you and I have come in contact with adenoviruses in the past. So what we may be boosting is a response to the adenovirus and not to the sequence we're interested in. So it's a very good approach, actually, to consider using two approaches from a prime to a boost. So I applaud them for going forward with that.
Now, that's only relevant for the viral vector approach. It makes no sense to have an mRNA vaccine as a first and to boost with an adenoviral, with a viral vector the second time.
I'm not totally convinced of that, actually.
Really?
I mean, to be honest, what you're trying to do is to induce a response to the sequence you're interested in. So with the mRNA, you're putting the messenger RNA in for the spike protein. So if you come in with a viral vector, especially a non-replicating viral vector, and get the expression of that protein again, in essence, you're boosting that response which was induced against the messenger RNA approach. Okay. So I wouldn't necessarily say it's a bad way of going forward. If anything, it's sort of an interesting approach and may actually be better than using this same approach twice.
I'll have to reconsider the advice I've been giving people then.
Oh, sorry. And that's sort of me looking at, how did we, with the HIV situation, how do we do that? And in a lot of cases, they would go in with one approach and boost with proteins. So you're going with adenovirus, boost with purified protein.
So to make this relevant for people listening, people have been asking if you get like the Pfizer vaccine and then you forgot which one you got, and you show up for your booster and you get like the Oxford shot as your booster and not the Pfizer as your booster, it might actually still work as a booster. Interesting. So. Okay. That I did not know. Fantastic. I'm learning things, people. All right. So I have in front of me the list of questions that people have submitted. And I'll go through each one, and maybe we can't answer them all, but we'll try. So the first one is, do you know if the federal or provincial governments have released priority lists of which subpopulations they plan to target with the first doses of the vaccine? I'll take a shot at this first.
Sure.
So every province has a slightly different approach. My understanding is that the federal government in Canada has distributed the vaccines according to whatever criteria they have, probably on a per capita basis, and each province strikes its own committee based on ethics and efficiency and strategy and so forth to figure out who gets it first. Now, in terms of subpopulations in Ontario, they're starting with the long-term care residents and healthcare workers, and then the elderly, and then down from there. The answer to your question is no, we don't know specifically after those three groups who gets it next. Will it be immunocompromised? Will it be caregivers? Will it be teachers? We don't know. And it's probably subject to change anyway, unless, Erling, you have information that I don't have.
Well, I'll just add to your commentary. When I was working with the Public Health Agency, one of the things I did was to support the National Advisory Committee on Immunization. The National Advisory Committee on Immunization has actually put together a preliminary guidance on key populations for early COVID immunization. So if you go on this, I'm not speaking to the public here, if you go on to just search NACI and look for guidance on COVID-19 vaccines, you'll actually see what the recommendations were. And there's a figure in that guidance, figure 1, which is a summary of the preliminary NACI recommendations on key populations for early COVID immunization. Lovely figure. Take a look at that one. It'll give you a good idea of what the federal government, at least what NACI, has provided as guidance to the provinces. And very much, as you're aware, healthcare in Canada is sort of it's a provincial jurisdiction, but the federal government obviously tries to influence what's going on to provide sort of universality across Canada. It's a uneasy relationship, as you probably are aware by all the discussions that have been going on with the ministers recently.
There's an old joke I always tell, because this is a podcast, can do what I want. I can tell jokes, why not? The UN has a contest, and all the various UN member states have to enter this contest. The contest is you write an essay about a topic, and the title of the essay must reflect the core values of your country. And the topic is elephants. And the French submit an essay called Elephants, a love story. And the British submit an essay called Elephants and their effect on the empire. The Americans submit an essay called Elephants, bigger, faster, stronger, better. And the Canadians, their essay is called Elephants, a federal provincial matter.
It's funny, actually. I usually come forward with, when we're discussing difficult issues, and I always say, well, okay, I'm going to state the elephant in the room.
Exactly. Save the elephant in the room.
And it's sort of interesting. It brings up Pierre Trudeau's statement about when asked what it was like to live so close to the US border. And I won't repeat the rest of it, but it had something to do with
elephants. Yeah.
Yes.
People can Google that as part of your homework, listeners, is Google Pierre Trudeau's comment about elephants. Okay, next question. If you get the vaccine, for how long will you be protected, and will it stop you from getting COVID or just reduce the chances? I suspect you meant reduce the symptoms. You want to take that one?
Sure. Right now, from the clinical trials that have been done, it looks like there's 95% efficacy in the two mRNA vaccines, at least, where that is a prevention of becoming infected, I believe, and specifically if you are infected from developing the worst type of disease. That is that you won't end up going into ICU. You won't end up having to go to the hospital. So if you are infected, it looks like that you'll develop mild symptoms. So for me, that's a win-win, to be honest with you. The fact that you have mild symptoms, the question is, will you be able to transmit the virus to someone else? That we're still not sure about.
Right, we don't know. And the trials weren't designed to detect that. They're designed to detect whether or not people would report symptoms relevant for them getting tested and therefore be tested for COVID. We will know better in a few months based upon the epidemiological signals. So as the test positivity rate changes in response to increasing vaccination penetration, that will tell us probably if we are indeed reducing transmission. Frankly, I expect that we probably are reducing transmission, if for no other reason that if we're reducing symptoms, lesser symptomatic people are less likely to transmit.
Yeah, I mean, that brings up sort of the early observations. Remember where we're saying asymptomatic people are actually transmitting? So there's still a question mark there in regards to virological things. I mean, I can bring this back to some studies I did in monkeys where you actually infected monkeys and looked at what happened to the viral peak. And that viral peak actually is very rapid after actually infection. 7 to 10 days, you have an amazing amount of virus hooching out there, and sometimes before symptoms. So I would say there's still a question mark.
Yeah,
but the whole idea of vaccination is that, and the holy grail is to reduce the amount of virus that circulates if you get infected and to have a short blip that's eliminated.
Yeah. Fantastic. Okay, next question. This one's maybe a medical question, but we'll see. I'm wondering about the vaccine's impact on people with autoimmune diseases and inflammatory conditions. 1, has it been tested in those populations? 2, for both people currently on on treatments like steroids and biologics, and those not on meds. I have sarcoidosis centered in my lungs and lymph nodes, but if agitated, it could go anywhere. So I'm concerned how my dramatic Tennessee Williams immune system will react to this.
Yeah. Do you want to take that one?
Yeah, I mean, that's, the answer is we don't know, because probably many of the exclusion criteria for the trials were to exclude people with these very conditions. So this is a conversation between you and your doctor to have. And frankly, I wouldn't be surprised if your doctor recommends holding off until we have more data.
Yeah, I'd agree with you on that one. And the other thing is the whole idea of vaccination is that we're trying to protect, in creating this herd immunity, ultimately, those individuals who are immunocompromised, those people under cancer therapy, for example, who will not have the same type of immune response as the rest of us. Individuals infected with HIV, for example, will not develop this type of immune responses. So by increasing the percentage of people that are vaccinated, we're going to protect those individuals.
That's right.
Yeah.
That's right. The next one is kind of similar. It's, would you recommend this vaccine to someone with long-term inflammatory conditions with the risks outweigh the benefits? But, I mean, what are these long-term inflammatory conditions? I mean, it's a big...
I mean, I'll be honest with you, age is a long-term inflammatory system.
Exactly.
And that's the problem, is in the elderly, actually, their immune response is more of an inflammatory immune response, which has been a big question mark in the development of these vaccines. You know, what are we going to be doing? We don't want to exacerbate an inflammatory immune response and send individuals into these cytokine cascades, for example.
Right. And obesity is a constant state of meta-inflammation.
Right.
So it, I mean, depends on what the cause is for your inflammation. Again, speak to your doctor. That's a medical issue.
Totally.
Okay. Next question. How can we trust a vaccine that hasn't been through all the phases? Well, I love that question, because clearly, go ahead. It has been through all the phases.
I jumped on that one right away when I thought, well, that may be dated as a question. Obviously, we've seen that there has been a lot of vaccines are actually in phase 3 clinical trials, which is the last phase before products are looked at by the regulators for licensure. So I would not say that they have not been through. In fact, the Pfizer vaccine is through and they're now licensed. The Russian vaccine has been through. They didn't go through all their phases of clinical trials. They were quite
creative.
Let's see, optimistic, maybe is the kindest way of saying it.
Yeah, I love the phraseology.
Ethics.
Yeah, I mean, and we're going through a phase 4 right now, the post-market phase. You know,
yes.
So I think it's important people understand that the speed at which this came, not to market necessarily, but into regular usage, is due to a number of factors. Number one is that scientists pivoted from working on Ebola vaccines to working on this, so the platforms are already there. Number two, we already had some experience with SARS and MERS, so the science was there. Number three, money. It's amazing when government underwrites the risk, how quickly we can get clinical trials ready to go. Number four, we pulled out all the administrative stops, like not waiting for scheduled meetings and things like that. Number five, manufacturing was started before the phases were complete. Number six, the clinical trials, the large trials recruited tens of thousands of people. Usually we recruit like hundreds for phase 3 trials, right? Maybe thousands, but tens of thousands. That means the speed at which we got data was accelerated. And as a result, things happened much faster. So no corners were cut. It's just that we spent a lot of money and effort to make sure things went faster.
The other thing that actually is quite fascinating is that the regulatory agencies were looking at the data as it was generated. Yeah. The companies were working closely with them. And for me, it's a game changer, without a doubt, in the field. So that shortened things as well. And I do not think we had a safety issue in doing that. If anything, the transparency was there.
Yes, absolutely. So next one, it's a numbers question. If a vaccine is 95% effective, does that mean it protects 95 out of 100 people entirely, or that there's a 5% chance of anyone getting infected? Want to try that one?
Okay. Well, I was going to throw it at your direction.
Oh, you're the sure. No, I'll take a shot then. All it means,
I would just say that 95% effective vaccine. Yeah, I mean, it means that 95% of the people that have been vaccinated, at least in the clinical trials that they looked at, were protected from one aspect, whatever they were looking at, either infection or disease.
That's right. I mean, specifically what it means is in this large population of people, 95% of the ones who got disease were people who didn't get the vaccine.
Yes.
Right? So it means maybe the vaccine has a 5% chance of not protecting you from showing symptoms. Again, not being infected, but showing symptoms. We don't know yet if that means not being infected.
Exactly. And to be honest with you, 95% effective vaccine. That's amazing. That's amazing. You know, in flu, we're looking at what,
40.
depending on the year, 10, 20, 40%
40
maybe.
the max, yeah.
Yeah. So, wow.
I mean, the cutoff that we're hoping to get for this one, as you know, is 50%.
Yes.
So that's incredible. And I think measles is like 92%. So measles is pretty good, but
Yes.
that's after decades of refinement.
Very much so.
So next one, what do we know / suspect about possible long-term effects, especially when vaccinating children? How can we make sure any risks associated with the vaccines that appear during the massive rollout are being openly shared with the public? Excellent questions.
Yes. I will answer it by saying that there is post-market surveillance being set up to look at adverse events. And in fact, Health Canada years ago set up post-market surveillance for a lot of the products that they actually regulate. So this is just another product that they will be looking at post-market surveillance. But to be honest with you, I mean, that the system is primed right now. If a company actually starts to see adverse events, they will respond to it. Litigation states that they cannot do any otherwise.
Right. And this is the most carefully watched biologic in the history of humankind. So there will be nothing hidden. The next question is kind of similar. It asks about mandatory vaccination in schools. And I want to say that the Pfizer vaccine has been licensed now for, not licensed, but approved for use of people 16 and older, which is odd to me because I think the trial data was for 18 and older. Okay. And right now, both Moderna and Pfizer are looking at testing it on kids as young as 12, but the data is not available yet. Therefore, we will not be seeing it used in children anytime soon. Probably not until next fall at the earliest, I would think. So the question of mandatory vaccination in schools, we're nowhere near there yet.
No, and it's also a question I would send towards the school boards, because ultimately they will be making the decision on what they require.
That's right.
It's being more, you know, are your parents vaccinated?
Yeah, and maybe the teachers too.
Exactly.
Yeah. So next one, are the mRNA vaccines safe for transplant recipients, and were they tested in any phase 3 trials? I assume on transplant recipients. That's from Margot. I guess the answer is no, they haven't been tested on transplant recipients. Is that right?
That's right.
Yeah. So we don't know the answer to that question. That's between you and your doctor.
But I would say again that if you look at the National Advisory Committee on Immunizations guidance, they do put guidance out in regards to transplant recipients and others in their general, you know, the Canadian immunization guide that they produce.
Okay.
So there's lots of advice being given to health professionals on what to do in specific situations. So I would say speak to your MD.
Exactly. And don't forget as well that you're weighing this against the risk of being exposed to the virus, right? And
exactly.
Yeah, and the virus is probably worse than the vaccine in all endeavors.
Well, have you seen that? It's a poster on vaccines. I actually use it in my lectures quite a bit, and it's pre-vaccination and post-vaccination with a lot of diseases. And the reduction of just being infected levels are, you know, 93 to 100%. But we don't even talk about what the secondary effects of being infected.
Yeah.
We're starting to see that with COVID. You know, these individuals that don't recover, they're long-term impacted. We don't know what will happen in the population of survivors.
I mean, most of the effects of a vaccine are due, correct me if I'm wrong, to two things. One is the other stuff that's in the formulation, the adjuvants, the stuff that's packaged in. And number two is your body's production of the antibodies themselves, which they would have produced in response to the virus.
Yes.
So I think it's unfair to cast the vaccine in a negative light if it causes you to be, you know, a little bit ill, perhaps, because it's going to be less so than the virus would have made you.
Yeah, viruses also, I mean, they're insidious. Some viruses actually will generate, or they have proteins encoded with them that will modify the immune response. And that's part of their survival strategy, especially some of the more complex viruses, DNA viruses, for example. So coronaviruses are probably one of the more complex of the RNA viruses. They actually encode some of their own error correction enzyme,
Ooh.
which is fascinating. So you don't see the same mutation rates that you will have with influenza, for example.
Yeah, which is a good question. Actually, I'm sure someone asked that down the road
Yeah,
here.
I think we do. Yeah.
So number 12, this is a good question. If there's uncertainty of how long people who have had COVID will have immunity after they've recovered, what is the certainty that the vaccine immunity will last longer? This is from Rena.
Good question.
Yeah.
We still don't know, to be honest with you. And, you know, I had a question from LinkedIn, actually, where people were saying, you know, why did Canada buy so many doses? And my response to that is, well, you know, being good Canadians, we like to have a balanced portfolio. Okay. And secondly, we don't know. We may have to boost in 6 months to a year.
That's right.
We're just not sure at this point. So
Yeah.
access to more than one.
I'll follow up though, that say that recovery from natural infection is highly variable because we don't know what kind of viral load you had, what kind of infectious dose you had, what kind of etc. So that's why there's so much variation in how much antibodies you produce and how long it lasts. Whereas a vaccine in general can be better calibrated. That's why there's a booster shot, to maximize the amount of antibody you produce and to make it last longer. So there's a higher probability that antibody and T cell production from a vaccine will be longer lasting than antibody production from natural infection. Would you agree with that?
I agree with that. And the other thing about it is that coronaviruses, I mean, they're not new to us. We have, you know, they will produce colds in the Canadian population. But we can be reinfected with coronaviruses after having a coronavirus infection fairly rapidly, like within a year, you can get another one. So we know that natural infection is likely not going to produce long-term protection already. And this is not new.
Right. So next question, what do we know about long-term safety? Well, I'm sure there's going to come up several questions here. I mean, I like this question because the answer is so kind of obvious, is you don't know until the time passes, right? Right. So we know nothing about long-term safety, except that, you know, we've been living with this virus for a year now, pretty much. And so we know what happens if you get infected a year later. So it won't be as bad as that, if anything. But we have post-market surveillance systems, and we'll be watching it very, very closely.
Very much so. And, you know, it's ultimately risk-benefit analysis. Right. You really have to look, what is the risk of getting infected and the long-term impacts of that versus what is the risk of having any adverse events that may actually, you know, continue lifelong. There may be, you know, adverse events. For example, we've heard with the rollout of the Pfizer vaccine in the UK that there were 2 people that actually got
allergic reaction.
Yeah, allergic reaction. Well, with allergic reaction, this is not new to vaccines. Every time I go in to get vaccinated, I have to sit down for 20 minutes afterwards because maybe I'll develop an allergic response. But the health professionals are there with EpiPens, with, you know, that will be given to me if I develop a reaction. I personally have never developed one of those reactions, and I've never seen anyone when I've been vaccinated en masse in a situation where I've seen any allergic reactions.
That's right. We should expect it, though. I mean, this is going to be rolled out to hundreds of millions of people. If the probability of an adverse reaction is 0.0001%, that still means hundreds, if not thousands of people are going to get a bad reaction. Don't let that spook you.
No, and also, as I said, health professionals are there with medications that can help you through that acute response.
That's right, if it happens, which is unlikely. Exactly. So the next question is also about immunotherapies. We covered that. Then is if you become infected with the virus between vaccine doses, can you still receive a second dose? Curious. So if you get the first shot and you haven't got the booster yet, and suddenly when you're waiting for the booster, you get infected, can you still get the booster?
Interesting question. And one of the responses to that is that the second virus infection, in essence, would be a booster.
Yeah.
In between the two.
Yeah.
So would you need to get another booster? Well, maybe you wouldn't need to.
It wouldn't hurt.
And also, if you give a booster soon after becoming infected, would you induce an immune response that actually may send you in, you know, another type of response, maybe an inflammatory immune response? Good question. We're not sure.
We don't know the answer. We can theorize, but we don't know the answer, unfortunately. Yeah, we'll pass on that one. So that one, what are, if any, worst possible, by the way, I'll back up for a second saying that's kind of why it's important that after you got your first shot, you don't drop all your defenses to go partying and things like that. You still got to maintain your distance and wear your mask and try to avoid exposures and infections.
And I'm telling people even after your second shot,
yeah,
you cannot, just go back and go into party time.
That's right.
It's very much going to be hold back. We have to make sure that the R naught is reduced. That is, the replication rate is reduced so that we're not transmitting to other people if we get infected. Or generally in the population, you're not seeing that transmission rate before we can start relaxing our basic public health prevention approaches.
The next question, this is a curious one. What are the worst possible unintended consequences of the mRNA vaccine technology? I never thought of it this way. Have you got any thoughts on what the worst possible unintended consequences might be?
That it doesn't protect you.
Yeah. I mean, I guess talking specifically about the mRNA approach, I see no downside to it, frankly.
No, I mean, to be honest with you, it's not a new approach.
Right.
It's a new regulated product. Okay. The initial use of mRNA was started in about 1990. It was very much looking at cancer research and a variety of other things. And they figured out that using messenger RNA with unmodified AUCG sequences, those are the nucleotides in RNA, would induce some type 1 innate immune responses. That is the responses that you get immediately when you come in contact with a pathogen. So they played around with it, and there was a group, and I'm blanking on the name of the individual, unfortunately, a group from Germany, collaborating with a group in the United States, that found if you modify the nucleosides, and specifically that U sequence, to a modified U, you would reduce that inflammatory immune response. So you'd have less inflammatory immune responses. So the approach has already been modified to make it safer. And this was done over the last little while. There's several publications. It has been tried, in several research and development sort of projects. I think they looked at it with Zika, they looked at it with influenza and a couple of other viruses as well. So again, not a brand new approach.
And we do have animal vaccines using this technology. I think pigs is a pig vaccine for it. It's been tried in rabies for humans too, right? So this is not the first time this has been used in humans. It's the first time it's been used en masse, deployed at the scale.
And regulated as a vaccine.
Yeah, and for me, the worst thing I can think of offhand is an ADE reaction, an antibody-dependent enhancement, right? Which seems unlikely. So that's when we have a suboptimal production of antibodies that in fact enhance the probability of infection.
Now that's fascinating as a commentary, because it's the exact question I asked to Barney Graham down at the National Institute of Health. He was instrumental in getting the sequence that was used for the Moderna vaccine. And he is an expert on ADE. He was involved in the respiratory syncytial virus vaccine approaches. I don't know if you're aware of this, but back in this, I think it was the 70s, there was a whole-killed vaccine attempted for RSV in children. And they found that in children that were vaccinated, they actually ended up with worse pathogenesis or worse disease if they came in contact with RSV than the control group, which stopped the vaccine completely. And this was thought to be an ADE response. That is, that having antibodies driven to specific epitopes within that virus or within the spike protein actually enhance the ability of that virus to come in contact with cells and infect individuals. Not something you'd want to see, obviously.
This stuff's amazing. I mean, I'm glad you're here to walk me through it, because it's well beyond my...
Well, I must say, I asked Barney Graham about this. I sent messages down immediately because I'm concerned about it. And the responses I got back said, yes, okay, he's dealt with this. He's really looked at it. He is very instrumental in the product development sort of group at the WHO. He supports them and specifically on RSV and influenza. So he's the right person for Moderna to have worked with. He just by chance actually works for Tony Fauci.
Oh, all right then.
Yeah.
Just drop that name.
Might as well, yeah.
Okay, next question is from Joseph. It's a good question. He's talking about the cold vaccines, the Pfizer vaccine, which is stored in extremely low temperatures. And he's assuming it's not for contamination control, which is true. It's to maintain the stability of the molecule. And he's assuming that's because heat can denature the protein. So he's asking, why doesn't the body heat itself denature the vaccine once it's injected into?
Okay, there's a couple of things there. The first, the Moderna and the Pfizer vaccines are both messenger RNA vaccines, so they're not proteins.
Oh, right, of course.
Secondly, messenger RNA, by its nature, can be chewed apart by RNases. These are enzymes that specifically destroy RNA. Those RNases are everywhere. I worked on purifying messenger RNA back in when I was a PhD student back in probably 1983. And the biggest thing I had to worry about was RNases, destroying the RNA I was working with. So I had to be super careful and store everything at -80 and make sure I use things that would destroy RNases. So part of the problem with the messenger RNA approach is how do you stabilize it? How do you protect that RNA? One aspect is cold. The other aspect was these lipid nanoparticles that are being used with messenger RNA. So they're taking, in essence, the messenger RNA and putting them inside fat balls. The lipid nanoparticles being fat balls, in essence, to protect them from any RNase destruction. But there's no RNase active at -70. Okay, it'll be totally out. Now, when you actually thaw that vial out to vaccinate individuals, you're going to bring it down to 4 degrees, probably on ice, keep the vial on ice for a bit, warm it up a bit before you inject it in someone. Generally, you don't want to inject something that's super cold into someone's arm. So you're going to warm it up, but in a very controlled fashion where you avoid RNase contamination, etc. So it's more what the environment could do to that virus, not what the body will do to it. The body will take those messenger RNAs, translate them into proteins inside our cells. Those proteins, some of them will be destroyed and presented as epitopes with MHC to the immune system, which will induce antibody production and T cell responses. That's a normal course of event. If you have a virus infection at all, messenger RNAs will produce protein. The proteins will be presented.
Are we not concerned about the RNase enzyme in the body once the vaccine arrives? It's injected.
I mean, part of the use, the use of those lipid nanoparticles, LNPs, is to get it into that cell.
Gotcha.
Brought into the cytoplasm to the level where the ribosomes can start translating it into protein.
Gotcha.
Now, once that happens, yeah, the messenger RNA is destroyed.
Interesting. Interesting. Okay. Next question. Will individuals with, oh, sorry, I skipped that question. This is from Miguel, who's, I think, a public health person. He's asking, what should we continue doing in terms of public health care once we get vaccinated? Well, covered this kind of, we don't let down our guard well until we have herd immunity. We got to continue wearing masks, continue distancing, continue to have limits on gatherings. Bit by bit, we can remove those things as the penetration of the vaccine is achieved, but that won't be for a long while.
Now, I talked to lots of people about this, and I've basically said, look, we have to continue doing this until everyone in the world has had a chance to become vaccinated, because viruses don't stay in someone else's backyard. And Canadians love to travel. So we're going to be exposed to anything circulating anywhere in the world. So that's right. Is going to take a while to happen.
Yeah. No one is safe until everyone is safe.
Yeah,
100%. Pandemic is global. We have to look out for everybody in all corners of the world.
Very much so.
Okay, so next question. Will individuals with previous positive COVID tests be bypassed for vaccines? Okay, this is I'll let you answer that, but first I want to say, I get this question a lot on Twitter, and here's, I tend to answer it this way. First of all, antibody tests in the blood donor population show that in Canada, at least, less than 4%, maybe even 1 or 2% of the population has antibodies for COVID already. So automatically, it's not a large number of people. Secondly, is we don't know if they are immune, how much antibodies they have present. So it doesn't seem like it's worth the juice for the squeeze to be testing people for COVID to have them bypass vaccination, especially since I don't see that vaccination can hurt them if they already have antibodies. What are your thinking on this?
I agree totally. If anything, it'll boost whatever immunity they have. So that's a good thing, to be honest with you. I want to have as many antibodies as possible.
Right, excellent. So we agree there. All right. How many people, this is from Robert, how many people in Canada will need to be vaccinated before we can see a decrease in cases? Okay, that's an epi question, so let me take a stab at that. Herd immunity, we think, is around 70%, and that's based on what the base reproduction number is. That's the average number of cases produced by an old case before we have these mitigation strategies like mask wearing and distancing in place. So based on what we observed in China, we think it's about 70%. Having said that, that's not the answer to your question. Your question was a decrease in cases, not herd immunity. And that's going to depend on how the vaccine is used. If it's first come, first serve, then really have no idea. On the other hand, if we're protecting the elderly and healthcare workers, then suddenly we have the ability to take those people off of the transmission board quickly. As a result, I can't give you a timeline or an exact number, but I will say we don't need to get to 70% before we see a reduction in cases, assuming that, in fact, vaccination does result in some diminished transmission. I would anticipate something much smaller than that, maybe 30 or 40%. That's my guess. Any thoughts on that?
I agree totally with that. And in fact, if we looked at influenza, that's what we're looking at. A vaccine that has, 10 to 40% efficacy. So if you're getting a half-care population vaccinated with a 95% effective vaccine, that you're probably similar to what's happening with influenza. But to be honest, the answer is we don't know yet.
Yeah. It's frustrating for people to hear, but that's the honest answer. We have to be transparent with our ignorance.
Yes.
This is a good question, and it's from Mary. After my 85-year-old parents are vaccinated, will they be able to shop without masks and have family over with confidence?
Well, my answer is nope. Yeah. Simple. No.
Yeah. Yeah.
And in fact, it's not just, we forget, it's not just COVID that's circulating. Right. We still have other viruses out there. There's other cold viruses. Influenza is still around. We're in the influenza season. Yes, by using masks and hand washing and everything, we've reduced the amount of influenza circulating in Canada. Yeah. But it's still there, and the elderly are impacted definitely by getting infected with these other viruses.
Exactly, and also keep in mind there is that 5% of people who will not be protected for whatever reason of the efficacy scores of this vaccine. I will say, my parents are in their late 80s, and when they are vaccinated, of course, I will encourage them to continue to wear masks and so forth. But I will not be angry if they see more of their family members, safely, of course, in the house. So this is an opportunity to diminish the mental health and the social burdens that people are feeling right now, being isolated. So I would never discourage them from seeing their loved ones with a bit more exuberance now, but don't be hugging and kissing people.
No, I agree. And it's increase your bubble, definitely.
Yes, exactly. Increase your bubble, but not your exposure necessarily, not until we've got herd immunity.
And you have to hope that the members of your bubble are understanding that if they get infected by increasing their bubble size, the impact on the elderly is there.
Right, exactly. So next question is from Julia. Do you know how much of the world will get the AstraZeneca vaccine versus the mRNA vaccines? Oh, excellent question. So my understanding is that the world's largest vaccine manufacturer, that's the Serum Institute in India, they are cranking out the AstraZeneca vaccine, and they intend to distribute that widely to the most populated parts of the world, like India and other parts of Asia. So I anticipate the viral vector formulations, including the Russian version, to have better global penetration than the mRNA ones, which require a more expensive infrastructure. Do you any thoughts on that?
I agree totally. It comes down to one of the big problems in the vaccine field, the cold chain.
Yeah.
Cold chain is always a problem. When I was arguing at the NIH about vaccine approaches for HIV, I said, look, if you have to go into certain areas of Africa where people may have to walk 10 miles to get that vaccine, do you think, number one, they're going to want to go for a vaccine that's unstable? And number two, come back for boosts every month? You have to look at your population and see what what would be the best for that population. And I think having a vaccine that does not require -70, but can be kept on ice would be a better approach. And in fact, if you can desiccate it, that is dry it out completely, and add water or something else at the site of vaccination, that is your best approach. Obviously sterile water, you don't want to be injecting some dirty water.
Yeah, I mean, and it's one of the reasons I'm glad that Canada has bought or made arrangements to buy so many different kinds of vaccines, because we don't know what kind we're going to need for what kinds of subpopulations as well, and hopefully we can donate our excess to the rest of the world when that time comes.
Agreed totally. And Canada actually has, it's our track record where, we do think of what's going on elsewhere in the world and very supportive of other countries.
So John asks a good question. I'll let you answer it first. Which COVID vaccine would you get for yourself? I have an answer for myself. I want to hear your opinion first.
Okay. My initial answer is, academically, I'd want to look at an approach that provides the best protection. And right now, the messenger RNAs would be those approaches. However, my realistic answer is whichever one I can get. I'm not going to be deciding that. Individuals elsewhere, I will put my arm out and I will take what I get.
Yeah, that's my feeling too. A friend contacted me recently. He has access to the Sinopharm, the Chinese vaccine, which is an old-fashioned technology, an attenuated virus. And he wanted to know if he should get it. Well, if it's being offered to you, I would get it. The safety profile looks okay. That's not an endorsement, by the way. It's based upon what I've read in the news reports. My feeling is what everyone has been okayed by Health Canada, that means the safety profile has been deemed to be acceptable, I'm more than happy to accept it. Having said that, if I have a choice, I love the mRNA ones. I think they're cool. I love the new technology. And the Moderna one, I have a slight preference for, only because of two reasons. One is, the cold chain issues are not as intense, and so I'm celebrating its distribution potential. But selfishly, it's got that one signal in the animal studies that showed signs of sterilizing immunity, and maybe they all do, but at least Moderna made that public. And so if it's got just a modicum higher chance of sterilizing immunity, I'll take it.
The other aspect about that one, and I agree totally, that Moderna would be my number one choice, because I know Barney Graham supported what the sequence is. It is not just a spike protein. It is a pre-fusion product, which induces better neutralizing antibodies in theory. So totally.
There it is.
I'm behind you.
I love validation from the expert. I love it. So this next question is actually from my spouse. She's cheating here. How'd you get your hair so nice? Because she cuts it. That's the answer. Okay, next question. How long?
I was going to answer genetically.
Maybe she's asking about your hair. It's true.
Well, I was, I sort of thought, but.
Okay. How long will it take to gather enough data to say something about length of vaccine effectiveness and/or whether COVID survivors can be reinfected? Is there evidence that there is more than one strain, like the seasonal flu? And will one vaccine address all strains? Wow. There's a lot going on there. I'll let you take the first shot, and I have some thoughts as well.
Okay. Enough data to say about the length of vaccine effectiveness? We don't have it yet. That's the quick answer. Whether COVID survivors can be reinfected, it looks like some of them can from the data that's coming out. Small numbers still, but some of them potentially can, because we're not sure, again, with them. And if we look at the coronaviruses, the other coronaviruses that cause colds, yes, you can get reinfection. So I would say that COVID infection isn't a guarantee that you can't be reinfected, which is one of the reasons why we're pushing the vaccine approach to induce a better type of immune response that may be longer term protected. Let's see, is there evidence of more than one strain? Yeah, we've seen there's other variants that seem to transmit easier, but this is not seasonal influenza. You don't have the same mutation rates with coronaviruses that you do with influenza. Influenza is fascinating. As a virologist, I love this virus. I hate it, what it does to people. But the amount of evolution that's going on, the genetic drift that happens every year, and those shifts that happen periodically that result in pandemics, absolutely fascinating. That is not the situation you have here with this virus. It's not the influenza.
That's right. Also, if the spike protein does change significantly, the mRNA technology is such that we can create a new version in 48 hours.
I agree totally. And that's the one exciting aspect of what's going on here, is that it may change how we do vaccinations, how we develop. Imagine an influenza vaccine that we didn't have to make a call in February for what we're going to give people in October.
Yeah.
If you could wait maybe to June, produce your vaccine and vaccinate everyone in August before we go back to school.
So a lot of the anti-vaxxer narrative around the flu vaccine is, some years they get it wrong and it offers no protection. That's right. Some years we do kind of miss the mark on it because you got to wait so long and take a guess at what the dominant strain is going to be. But as Erling just said, with the mRNA technology, we can get it almost bang on every year from now on if we use this technology.
Exactly. The other thing was, the initial influenza story about getting it wrong, that was usually, in the vaccine, we have usually 2A viruses that are the dominant viruses circulating in February. And one of 2Bs was the original approach. These trivalent vaccines that, in fact, the high dose vaccine for the over 65s is still a trivalent vaccine. So if we make the wrong decision on which B virus is going to circulate, well, there's no protection from the vaccine if you're infected with a B virus. Fortunately now, for anyone under 65, you're getting a quadrivalent vaccine, which has 2As and 2Bs. So there's better protection. I remember hiring health economists to look at this to try to convince the provinces that, that extra investment makes sense.
Right.
Reduction of disease. But it was a hard sell.
Well, thank you for doing it. Okay, next question. Good question. Is it cold when administered? Do you feel cold when injected into your arm? I don't have an answer for that, except I don't think we have cold proprioceptors inside your arm, so you probably wouldn't feel it.
Exactly.
Yeah.
I mean, the thing I feel always is the needle going in.
Exactly, yeah. So it might be cold, but you wouldn't feel it.
Exactly.
Okay, next question. Should we wait to get the vaccine or should we get it as soon as it's available? Get it as soon as it's available.
Times 10.
Yeah. If it's offered, take it.
Exactly.
That's again, neither of us are clinicians. We're not advising you. My personal opinion, if it's offered to me, I'm just jumping on that and getting it right away.
And I'm going to add, I don't work for the government anymore. I'm retired from Health Canada. It's been over a year. I actually now can speak publicly without having to worry about my conflict of interest. And second, I don't work with industry. I work in an academic institution. And as an academic, I don't agree with everything. The whole idea of being a scientist is we don't agree with absolutely everything. We will question, we will look at everything, with a fine-tooth comb to see, is there any issues here? That's what I love about being a scientist, is that I never accept anything without examining the data, teasing it apart, looking at it, talking to people, chewing it over. The best place I've ever done science is in a pub in Cambridge, UK.
Really?
Fantastic. Putting together ideas, throwing ideas apart, obviously over a beer.
Yeah, I'm just focused on the pub thing. I haven't been in a pub in so long.
Terrible, isn't it?
I'll say too, I haven't gotten any checks from Bill Gates at all. In fact, if anything, I've given him money for using his products. So next question is a good one. With the 2-dose vaccine, if you have side effects from the first dose, will the side effects from the second dose likely be better, worse, or similar?
You want me to answer that?
I have some thoughts on it, but you take it first shot.
Okay, if the side effects are an allergic response, then I would say likely it would not be a good idea to get the second one unless you enjoy anaphylaxis. Okay, so I would say that obviously in that situation, you'd be speaking to your doctor. whether it's advisable to get the second dose. The whole idea of trying to vaccinate as many people as possible is to protect you as an individual, because you may not be able to get your full regimen of vaccines because of these side effects that could be significant. We don't know yet. We're not seeing them, but when we vaccinate billions of people, we're likely going to see some adverse events.
Right. Yeah, it's going to vary a lot. But for the small things like fever or a little fatigue, if experience with other vaccines is an indication, it would probably be the same or less. I had the shingles vaccine 2 years ago that I talk about endlessly, the shingles vaccine. It knocked me out on my ass for 2 days in bed. I have never felt so untoward in my life. And I was terrified to get the booster, and I understand the science. And I slinked in there and I took 2 days off work knowing this is going to be really hard, and nothing, it was fine. So the booster had zero effect on me in terms of side effects, whereas the original one just knocked me on my butt. So I think most people, I say this anecdotally, having asked many people, those who've had bad side effects the first time get none the second time. But again, your mileage may differ. So
exactly. I mean, there's another thing about it too, is that I tell people, if you get a sore arm after vaccinating, it means it's working.
yeah.
You're getting a local immune response, an inflammatory response there. Your immune system is working. Fascinating. When I don't get any sore arm, I question whether the vaccine actually took.
So this next question is from Joseph. It's a good question, and I actually asked my physician spouse about it as well, and she has an answer if you haven't got one. He asks, why are vaccines typically administered intramuscularly instead of intravenously?
I'd be interested in hearing what your wife actually said.
Well, she didn't know for sure, but she theorized that it's because things that are given intravenously are more likely to be filtered out by your kidneys quickly, whereas intramuscularly they'll seep into your bloodstream and are more likely to find purchase in your system before being filtered out. That's her theory.
There's another little thing. I mean, intravenously, obviously, it spreads all around the body very quickly. Intramuscularly, you're going to be presenting that antigen or messenger RNA to antigen-presenting cells locally, which will induce that immune response that you want to develop locally. So you'll get, for example, if you administer something for a respiratory infection that comes through your nose, if you took a vaccine that's administered by aerosol, for example, in the nose, you'll develop local immune responses, which will protect you better. So that approach. In regards to this, it's basically getting it into the antigen-presenting cells as quickly as possible and inducing those responses we want to see, which will circulate, go to the lymph nodes if it's a T-cell type approach, or you'll get antibodies expressed all over the body.
Well, your answer is better. I like your answer.
I've been talking the talk for a while.
Yeah, I'm so glad I got you on this podcast. So glad. Last two questions are actually the same question, so I'm going to combine them. It's from partly from Mike and partly from Jordana, and essentially asking, how can we trust that it's safe if it hasn't been fully tested yet? And Mike's asking, how can we reassure people that no safety steps were skipped? Same question. And the answer, of course, is that no safety steps were skipped. None. It's just the speed is the result of administrative oversight of making things more efficient and throwing a lot of money at the problem. Do you have anything to add for that?
I agree totally. The only one I would say maybe not would have been the Russian vaccine.
Yeah,
I love the name, by the way. It's Sputnik 5.
It's Sputnik V apparently.
V, okay,
vaccine.
okay. Well,
yeah.
I'm saying 5 because I was born in October 1957 when the first Sputnik was sent up.
Whoa.
So I have sort of a certain liking for the name, but I'm also concerned about the fact that they approve those products, and they're not the only ones doing it. That there's another product that they are generating that has been approved in phase 1 clinical trials. So I'm a little concerned about what they're doing.
As am I. I mean, frankly, if you skip certain steps, you're not transparent about certain steps, I can't be confident you're transparent and have proper oversight over additional steps. So it's consistency of approach that's important to me.
It's sort of interesting. I have some Russian friends that have asked me point blank, would I take their vaccines? Interesting question. Interesting question. until I saw the safety profiles, I would not be taking that vaccine.
Yeah, I feel the
Because
same
I'm
way.
not sure who generated, how they generated, how the purifications were done, who did it, where was it done. I know that the infrastructure in Russia is not what it used to be. So I'd be a little concerned about what was being done.
Right. I mean, technologically, it looks safe. It's the same as the Oxford AstraZeneca approach. But you're right, it's the impurity aspects of it. It's the details that matter.
Now, the AstraZeneca used the chimpanzee coronavirus. I thought the Russian one was a human adenovirus construct.
Kind of like the CanSino one, right? They also use human.
Yeah, exactly. So, I'm a little concerned by the fact I like the chimpanzee adenovirus approach, because guess what? The last time I figured out, I have never come in contact with a chimpanzee. I stay away from them. And secondly, likely I'd never come in contact with a chimpanzee adenovirus. So I don't have pre-existing immunity to that vector, which is the beauty of that approach.
Right. Well, those are all the questions, Erling. That's fantastic. I learned a lot here.
I enjoyed it.
So let's end it there. Thank you and if there's any follow-up questions from the public, I would love to have you back on, if that's possible.
Great. Yeah, no, it'd be lovely. I enjoy this. Maybe we can, I don't know what frequency you'd consider doing it.
Sure. And next time, of course, I'll ship you some beer so we can sort of drink at the same time.
That's lovely. Definitely.